Let me begin with a question. What if an impurity in your drug product is not coming from outside, but is actually formed from the drug molecule itself? This is exactly what regulators discovered during the nitrosamine crisis. Today, we'll explore nitrosamine drug substance-related impurities, or NDSRIs, what they are, how they form, why they matter, and how we can control them. This knowledge is essential for anyone working in pharmaceutical development, quality, or regulatory affairs. The story begins in 2018, when nitrosamines like NDMA were detected in drugs such as sartans, ranitidine, and metformin. These compounds are classified as probable human carcinogens, which immediately triggered a global safety review. Initially, regulators focused on nitrosamines as external contaminants, coming from solvents, reagents, or raw materials. But soon, the focus shifted. They realized that some nitrosamines were actually forming from the drug substance itself. This discovery introduced a new category of impurities, NDSRIs. So, what exactly are NDSRIs? NDSRIs are nitrosamines that are structurally related to the active pharmaceutical ingredient, or API. They form when a drug contains a vulnerable secondary or tertiary amine group. When this amine reacts with a nitrosating agent, it creates a nitrosamine impurity. Unlike external contaminants, these are modified versions of the drug molecule itself. This makes them more complex to detect and control. For NDSRIs to form, 3 key components must be present. First, an amine source. This comes from secondary or tertiary amines within the drug structure. Second, a nitrosating agent. Nitrites can be present in excipients, water, solvents, or raw materials. Third, the right reaction conditions. Acidic environments during manufacturing, storage, or degradation can promote nitrosation. When these factors combine, NDSRIs can form. NDSRIs are critical because of their safety impact. They are genotoxic, meaning they can damage DNA and potentially increase cancer risk. Because toxicological data is often limited, regulators take a very cautious approach. They set extremely low acceptable limits, often in parts per billion. This means even trace amounts are important. That's why controlling NDSRIs is a top priority. Regulators expect companies to take a scientific and systematic approach. This begins with risk assessment. Companies must evaluate whether their drug structure or process can form NDSRIs. Next comes testing. Sensitive analytical methods like LC-MS are used for confirmatory detection. Then comes control. Processes and formulations must be modified if needed to prevent formation. Finally, compliance. Scientific assessments must be submitted to regulatory agencies like FDA, EMA, and ICH. In practice, risk assessment starts with structure review. You must check if the API contains secondary or tertiary amines. Next is material audit. This involves evaluating nitrite levels in excipients, reagents, and water. Finally, stress studies are performed. Forced degradation studies help determine whether NDSRIs form under stress conditions. This proactive approach helps identify risks early. Analyzing NDSRIs presents several challenges. First, detection limits are extremely low, often in parts per billion. This requires highly sensitive methods. Second, structural similarity between the API and impurity can cause interference. Third, sample preparation is critical. Improper handling can even create nitrosamines during analysis. Finally, advanced instrumentation like LC-MS or high-resolution mass spectrometry is necessary. Fortunately, NDSRIs can be controlled with the right strategies. Material quality is essential. Nitrite sources must be minimized, and raw materials carefully controlled. Process optimization also helps. Controlling pH and manufacturing conditions can prevent nitrosation. Formulation design is another key factor. Choosing the right excipients and packaging can reduce risk. These steps help ensure product safety. Let's conclude with the key message. To control NDSRIs, you must know your molecule, understand your process, and identify your risks. Nitrosamine testing is not just a regulatory requirement. It is a scientific responsibility. Because ultimately, our goal is to ensure patient safety. And that begins with understanding and controlling impurities like NDSRIs. Thank you